Well, I think we can get started. So yeah, hi everyone. Thanks so much for coming to our first AMPD public webinar. So to those who have joined us for our internal webinars, welcome back, and to those who are new, welcome. We're excited to share this with you and also get your feedback. So here's our agenda for today. So a quick introduction in the first 15 minutes with an introduction into AMPD for those of you that are newer to the program. And then we'll jump into going through the notebook that looks at APOE status across the AMPD data. And then at the end, we'll have a Q&A and discussion. And so we've received a lot of interest in these sessions, hence we've expanded them, and then this is our first public webinar. So in addition to, thank you for coming and attending, we would also really appreciate your feedback. So with this survey link that I'm just going to post it in the chat. So if you could just fill it out as you're I guess, paying attention, that would be really helpful. It would be really great to get your feedback and just make these as useful as possible in the future as well. So thanks so much for coming. And yeah, we'll get started. My name is Victoria Dardov. I'm a principal data scientist at Technome, and I'll be presenting the notebook today. And we also have 3 other team members on the call. We have Willie, Dave, and Barry. Willie, if you want to.
Yeah. Hi, everyone. I'm Willie. I'm a solutions engineer at Verily Life Sciences. I've been working really closely with Tara for the past 3 years as the solution engineer. I've also been working really closely with Technome and AMPD for the past few years, developing notebooks, not only in the proteomics workspace, but in several of the other workspaces available too. Pass it over to you, Dave.
Sorry, trying to get to my microphone button. I'm Dave Ismer with Technome. I'm a senior engineer, and I'm working on AMPD as the data operator. So managing a lot of the infrastructure, doing some data processing, data harmonization, and assisting users in getting access and using the data.
Thanks, Dave.
Sure.
Barry? I saw Barry on. Yeah, Barry's on here. Barry, did you want to introduce yourself real quick? I think Barry might be having some technical difficulties because I did see him on here, and he is on here. So we'll let him introduce himself when he's able to. So with today's format, because we do have part of it that could be a hands-on session if you already have tier 2 access, and I'll get more into that in a little bit. If you want to participate in the hands-on section and you have access and you're having any issues with running the workspace, Willie, Dave, or Barry can help you. They can bring you into a breakout session and help you with any issues you might be having. In addition, if you have any questions, you can put them into the chat. And then Willie, Dave, or Barry will be able to help you with those questions. And then this way it'll keep the workflow, or it'll keep the webinar flow consistent and won't interrupt that. So yeah, Barry, I see you on there again. Did you want to introduce yourself?
I am. Yeah, for some reason I couldn't unmute myself. I'm Barry Landon. I'm a solutions architect with Technome. I'm also the data coordinator for AMPD. And today I'm running support for this webinar.
Awesome. Thanks, Barry.
Sure.
So with that, we'll get into the introduction. As most people are familiar with Parkinson's disease and AMPD, I'm going to keep it brief, but I did want to do a little bit of background for anyone who might be not familiar with AMPD or Parkinson's disease. So what is Parkinson's disease? It's a neurodegenerative disease that's caused by death or malfunction of dopaminergic neurons in the substantia nigra, which is essentially a really fancy way of saying there's a decrease in dopamine signaling for either either caused by death of these cells that are releasing dopamine or malfunction of these cells that are releasing dopamine. And it can lead to a lot of different symptoms, including both motor and non-motor impairments. And currently, within Parkinson's disease, there's no cure. And then there are current treatments that are prescribed to patients with Parkinson's disease that are aimed at treating the symptoms. And so theoretically, while the disease is still progressing at a consistent rate, you are able to decrease the severity of some of the symptoms with treatment, but the treatment itself does not change disease progression. And so this is the case with a lot of neurodegenerative diseases. We just don't have cures for them, and the treatments aren't really targeting the root cause of the disease. And so with that, in order to kind of help identify and validate the most promising biological targets for therapeutics, the AMP-PD partnership has kind of taken form. And then over the past, I guess, 4 or 5 years or so, it's been around. And it is a partnership between industry, nonprofit, and government aimed at identifying and validating these most promising biological targets for therapeutics. And the way that they've done this is through having a wealth of data that's available. And so with that, I'm going to go to our data dashboard on the AMP-PD website. So this is a really great resource. The AMP-PD website has a lot of information, so feel free to browse it on your own time. But this data dashboard is really useful in helping understand just how many samples there are within AMP-PD. So within AMP-PD, there are 8 cohorts provided by different data providers. And this amounts to over 10,000 total participants. And so within that, there are Parkinson's disease case participants, and there's also control participants. And then there's also participants with other diagnoses. There's a lot of really nice visualizations kind of outlining the demographics, biological sex. Here are the most common diagnoses that are within the data set, race distribution, age distribution. And so all of this is, I guess, focusing on the demographics of the patients. And then we're getting into just how many samples have whole genome sequencing, how many have RNA sequencing, and how many samples have proteomics performed on them. And so within the AMP-PD data set, there's a lot of clinical data, and then patients with clinical data also have some form of either genomics, transcriptomics, proteomics, some combination of the 2 or all 3 of those omics available for CSF and plasma samples from these cohorts. And so there is a lot of data out there, and we're just going to be focusing and narrowing in on one subset of data in today's presentation. So I just urge you to look at this. If you're interested, you can click on different cohorts, and it'll tell you how many participants are within that cohort and give you the same type of breakdowns for each of the cohorts that you click on. You can also obviously click on multiple. And here we have the PPMI cohort as well, almost 2,000 participants. So you can see the breakdown again for the demographics, and then proteomics is available for this cohort as well. What's also really great about this data is that there are different collection time points. So there is a baseline visit, and then there are separate collection time points for CSF and plasma throughout the studies. So you can do longitudinal analyses if you are interested in something like that. With that, I'll go back to here and go to the full view. And so this data dashboard is available on the website. And in order to make full use of all of the omics data that's available, there are a wealth of AMP-PD community resources, one of which we'll be talking about today. We do have data explorers and data visualization tools that are built and are available for use. We also have a lot of workspaces and notebooks that range from basic mechanics and getting started and where is the data, all the way to different omics analyses, so proteomics, genomics, transcriptomics analyses. There's also workspaces and notebooks that start to integrate different omics. And then there's data production workflows and QC analyses as well. So if you wanted to run workflows on, let's say, RNA-seq, those are available as well. And from the community, we have more than 50 notebooks that are planned and underway that are being built by the AMPD community. So we're very excited for those. And in addition, we're using this webinar series to showcase some of these community workspaces that are being built. And so we're very excited to share these with you and get the word out about the AMPD data and also the AMPD resources. And so with that, we have 3 more webinars that are scheduled, one towards the end of May, one in June, and one in August, with more to be scheduled as more of the community workspaces become available. And so with that, before I get into running Terra workspaces and doing some more of the demo work, does anybody have any questions? Okay, I will take that silence as there is no questions, and we'll keep going. So this is what the Terra homepage looks like, and I'll navigate there and start doing it live in a second. This is what the homepage looks like, and when we log in and look at our dashboard, this is the one workspace that we'll be using. And so if you have tier 2 access, I'll be demoing this in a second, but this is the workspace that we're going to be using. It's hopefully clearly labeled with AMPD webinar and today's date and ApoE genotype. So that's the one we'll be using. And within this workspace, there is one notebook, ApoE case study, which is the notebook we'll be running today. And so with that, I'm going to switch over to the actual workspace. So here's our workspace. So my view is a little bit different than yours, just because I have a lot of workspaces on mine. I have a lot of access to other people's workspaces too when they clone any of my workspaces. So that's kind of why mine seems a bit crowded, but yours should seem a lot cleaner. So actually, let's just start from the beginning. So from Terra, here we're going to log in. So if you, I'm actually logged in, but you can just log in. You need to have your account set up through Google to be able to log in. And if you have tier 2 access and have used Terra before, then you should be familiar with that. Within our workspaces, these are all the workspaces, and I typically sort by the date. We can see that Willie, who's on the call, has just cloned his. And so we have this one right here. So this is the one we're going to be using. And so just to make it easier to find, we can just type in webinar. So here it is.
I'll also post a link to it in the chat.
Oh, thanks, Willie. That's very useful. So yeah, so Willie, so when you click on the link, you're going to be seeing this, and I'm going to start going through the different parts of the notebook and workspace once we clone the workspace and get the cloud environment going. So the reason we want to do that is it does take a little bit of time for the cloud environment to start going. And so we don't want to just sit here in awkward silence waiting for 5 minutes. So we'll clone it, and then I'll do the walkthrough, and then we can get back to running. So on the right-hand side, you should see this little snowman icon. If you click on it, you'll be able to clone it. And you can rename it. I like to put my last name in front of anything that I clone just so I can easily find it. And if you have access to this workspace, you should also have access to this AMPD release 3 stage, which is the billing project you're going to use. And then that's pretty much it in terms of setting it up. So just rename it to something that makes sense to you. Make sure you're using the release 3 stage billing project for these webinars. And then you can click clone workspace. And so now it'll clone. It'll probably take a few seconds to clone. And once it's cloned, here we go. We can set up the cloud environment. So we can click on this little gear icon right here. And we can use the default environment. The default environment works well for this, so you can create. And so now that's going to be creating over here. And while that's creating, we can start doing some walkthroughs of what the workspace actually looks like. So this is the workspace dashboard. There's essentially info about the notebook and the workspace. So nothing too exciting, but it does show you, if you forget how to clone, it'll tell you how to clone it. If you're new to the Terra environment, it'll tell you how to find the how-to guides. If you're interested in where's the data, this will lay out all the data that is currently available through AmpPD and the Google Cloud Storage locations. Also the Google BigQuery locations for all the data that's also been previously released. So we have all of these locations here, and let's just say we're interested in seeing this one. So we can click on it. And if you have access, you should be able to kind of go in and see what the data looks like. So I think it was this one right here that we had clicked on. And so you can see all of the different data sets that are available in BigQuery in this specific data set that is linked to from here. Within this, we also have files and workspace data. The workspace data is copied over, and it has all of the locations for where the data is in Google Cloud. Is it possible to zoom in? Yes, I can zoom. All right, I have like a big screen, and yeah, is this better? Okay, sounds good. Sorry about that. So yeah, this is the workspace data, and it just shows, what we're calling this and where in Google Cloud Storage this is. And then files, if you run anything in the virtual machine here and save it, then it'll show up here. And then this is our notebooks, our notebooks. So this one is still running, so it's going to take a few more minutes for this to copy over. That's totally fine. The cloud environment is still starting up, so this is going to take a little bit more time. But all the notebooks are within this notebooks frame. And then you also have workflows. So if you were with us in the last webinar, we did go through a workflow and kind of how to set that up. So workflows are very powerful. So this is, if there were a workflow in here, this is where it would be. And then this is where the job history for the workflow would be. Okay. And so it seems like my cloud environment is running, but the notebook is still copying over. So we'll just need to wait a few more minutes for this.
And if you refresh that page, Victoria,
oh, so
it's still being copied.
Okay, I'll refresh it and see. Yeah, I guess it's still copying over for this. Are other people still copying theirs over?
I did see it for a little bit, but after refreshing, after a minute or so, it copied over.
Mm-hmm. Okay. I wonder if I started the cloud environment too early. I don't know. Is that a thing?
They're copying over the notebook and starting the cloud environment are unrelated.
Okay, then.
So
no. So, okay.
If this is taking a while, an alternative you can do is go back to your main workspace since you're the owner of that.
Yeah.
But it looks like.
So yeah, plan B, we'll get this cloud environment started, making sure that it's running while this is copying over, just in case it takes a little bit longer. It seems like it's taking a little bit longer than normal, because usually it'll copy over very quickly, at least in my experience.
Perhaps while we're waiting for the notebook, would it be worth going over the workspace dashboard and mentioning what's written there?
This one?
Yeah.
I did already go through it. Does anybody have any questions, I guess, related to the dashboard and any of the data and where it's stored. Anyone? Okay. Well, it seems like the original one is working okay, so we can get started with that. And what I did,
Did I see the if you refresh this, still no notebook?
yeah, this is the copy. Oh, there it
There
is.
it is. Cool.
Yay.
Just needed a minute.
Yes. Okay. So now that we're good with that, so we can, so once we're in here, we can click open or playground mode. And so there's two ways to edit these notebooks. One is playground mode, in which you can edit, but it won't save any of your changes. It won't auto-save any of your changes. And then there's a manual way to go in and save it. And then you could just click open if you plan on opening and editing and saving. So I'm just going into playground mode just because I don't plan on editing anything. And so what we can do now, now that it's open and the cloud environment's going, and now we can edit our notebook, we can actually just go to all output and clear. And so what this does is, because I run this workspace in the I guess the master copy before, it is populated with all this data. And so I want to get rid of that. And so if I go to cell and then all outputs and clear, it'll clear everything. And then we can start running things one by one. And so I'll start going through this now. So the first part of all the Terra notebooks, including this one, is setting it up. And so if you're doing R, you're going to set it up this way with loading the libraries. If you're doing it with Python, there's a different way, I guess a different specific set of code to load things and set up those notebooks. But this is for R. So we're just going to click on the cell, and then you can actually press the run button. And then same thing here, right? Set up the utility functions. So these are just basic functions. These are all standard across most of the notebooks that I've written. So there's no need in, I guess, editing those. This notebook globals points to our workspace attributes and kind of where our data is. So this could be edited if you wanted to point this to other AMPD data that wasn't listed on here. And then same thing. So now that we have, now that our workspace attributes have been loaded and it kind of knows where our data is because we've told it where our data is, we can start looking at what BigQuery tables we have available. And so here I just have some code that lists the BigQuery data sets that are available. And so this is for a tier 1 release data set. And so this is all of this clinical data that is currently available in BigQuery. So we do have a lot of clinical data that's available. This is the targeted proteomics data set that is currently available. And so we're going to be using some of this, and we're going to also be using this APOE mutations table that's in here. And then this has other tier 2 data. And then this has, this is where our transcriptomics data is. And so these are the transcriptomics BigQuery tables that are also available. So within this notebook, I did not use any transcriptomics data just because for, I guess in the interest of time, it did take a while to load the data and work with it. So if you're really interested and you have access, you can start playing around with the transcriptomics data that's already previously available and just substituting, I guess, the transcriptomics data tables for the proteomics ones that are later on. So now we're just going to read in our BigQuery tables here. This is just setting up all the variables. And then I like to do a lot of different checks within the data. So I like to do a lot of these print functions and a lot of these summary functions that will essentially show me what the data looks like. So here I'm printing the unique study ID or the studies. So the 8 different cohorts that are available. So this answers the question, are all 8 cohorts in here? And then a summary of the participants. So how many unique participants are there? So there's 10,772, which matches the number that's on the AMP-PD website. And then I'm going to start merging tables. So what I'm actually going to do here is just click on this set and just run all of these, and then we'll go through them. So I'm calling this demographics table. So I'm making this new table, essentially merging the demographics data and the participants data. And then so that I'm adding in extra columns. So I have within here, I'll have participant ID, study, and so on. And then also all this demographics information as well. And I did that twice. I then also want to look at a summary of the mutations table. So here is a summary of the mutations table. There are 10,418 participant IDs within the mutations table. And then these are the columns. So there's GBA mutations, LARC2 mutations, alpha-synuclein mutations, and then just has known PD mutations and whole genome sequencing. I then merged the ApoE table with the mutations table to get this merged table that has both ApoE and other mutations as well. For this particular notebook, I do only use the ApoE, but if you were interested, you could go in and look at the other ones as well. So now I merged this mutations table that I had just made with the demographics table. And this is a sample of what the data looks like. Because this is a public webinar and we're not displaying any individual participant level data, we've removed all of the, sample identifiers. So you're still able to see kind of the structure of what the table would look like, but you're just not able to see, which patient it belongs to. But this is what the table looks like. And then we also have, I do a lot of these summary ones, especially when I'm merging things, just to make sure that everything's looking right. And I have all of these columns properly merged. And so based on this summary info, there are some things that we can further investigate. We could look at ApoE genotype. There's also ApoE4 status. And there are also other mutations as well. And so moving further into the notebook, I do end up choosing the ApoE status for further graphs. But before we do that, I did want to graph how many participant IDs are associated with each study. And I think I do need to zoom out a little bit just so that you can see the full pie chart. Yeah. Too much zooming, too little zooming. Okay. Well, it seems like I can't zoom perfectly enough. I'm going to try and fit this into the frame. But this is a pie chart just showing of the participants. This is the breakdown of the different cohorts that are in there. We do see that a large chunk of our participants are made up by this LBD cohort. We do also have a lot of PPMI and PDVP patients. So this is just a breakdown by cohort.
Hey, Victoria.
Yes.
If you wanted to look at that pie chart more closely, another option is to right-click and open image in new tab. Something we've learned in one of the past webinars.
Yes. Thanks, Willie.
Mm-hmm.
Willie is our resident Terra expert, so he's been helping me a lot. Thanks, Willie. Also something that I found out was you can also save image as or copy an image. So I had always written code to export all these graphs, but you can simply right click and save it, which is actually really nice. We then asked the question, how many participant IDs have known mutations, known PD mutations in whole genome sequencing? So this just graphs one of those column, so has known PD mutations in WGS, we do see that about 20% or so have known Parkinson's disease mutations in whole genome sequencing, and over 8,500 patients, participants do not have known PD mutations. Next, the question is how many participant IDs are associated with each APOE genotype? And so for this one, we graphed another pie chart looking at the different APOE genotypes. And so I'll just open this up in a new tab. So we have currently 6 APOE genotypes going from E2, E2 all the way to E4, E4. And then there's the different heterozygous ones as well. And so we do see that a majority of the participant IDs have this standard E3, E3 APOE genotype, which is the most common. There are some that are E3, E4, and we have some E4, E4 over here. We do also have some E2, E2, and E2, E3. And I included some links down here, just going over what APOE is and the different genotypes and what they mean. So E4 is more highly linked to neurodegenerative diseases. So that's partially why we're interested in APOE. And so what I'm doing now is, so now I had the question of let's separate it out by APOE genotype, but also look at the different cohorts. And so with that, I separated out and summarized the data and grouped it by APOE genotype and study. And then I was able to make these graphs. And so I'm going to open this in a new tab and just zoom in. So we have the different, so I will be zooming into this, but we do have the different cohorts here, and then the number of participants. And so, and then at the bottom here, we do have the different APOE genotypes. And so you can see kind of how the numbers vary by genotype also across the different cohorts. So you're able to see that. And yeah, this is kind of like a really big figure, but it does have a lot of information and separates out each APOE genotype by the cohort as well. And like I said, there's some links here included on APOE and stuff like that. So you're more than welcome to click on these and read a little bit more on APOE. So the next set was what happens when we merge APOE with proteomics and case control. And so what I do is here I'm merging the case control with the previous clinical data table that I had made. And this is the summary of that. So now we can see, you know, there's all the clinical data. And then at the end, we've added these case and control and diagnoses columns. I then merge this with proteomics data and then end up with a really large data table. So just in, you can see that there are 38 columns within this large data table. And it includes participant IDs, but it also includes things like NPX, which is the normalized protein expression, which is what we're really interested in for the proteomics data. And then this is an example of what the data set looks like. So this is only showing the first 16 columns, excluding participant IDs. But we can see we have, you know, omics, we have some standard metadata information about the targeted proteomics assay. And then we also have our NPX here, as well as some QC information. And then that's followed by a lot of other columns that are listed here, but not included in this example table. Now, what is the NPX distribution of participant IDs based on APOE genotype? So we looked at the NPX distribution, so the normalized protein expression distribution for all participant IDs, and grouped them by APOE genotype. So that's right here. So we can see these are the 6 APOE genotypes. And we can see the NPX distribution is fairly similar for all ApoE genotypes. And then if we had separated this out by case and control, we can see here, this is ApoE genotype NPX distribution across the different ApoE genotypes, but we've also separated it out by case, control, and other. So this lets us answer the question, is the NPX distribution different for any of the ApoE genotypes, and is it different between case and control or other for each of these genotypes? And then we can start graphing and looking at those numbers of ApoE3, E4, and seeing if there's more in case or control. So here we've grouped it by case, control, and other, looking at the different ApoE genotypes and just the number of NPX samples. And so if you're looking at the scale thinking it looks really high, so for each participant ID, there are multiple time points. And for each of those participant IDs with multiple time points, there's multiple proteins that are also being detected. And so this number accurately represents all of the UniProt IDs that are being detected for each sample for each time point. So we're able to look at that this way. And then just to answer the question of, within, we can look at this number, but, are the number of E3, E4 cases in case versus control really much higher, or do we just have a higher number of samples? I didn't actually do the percentages on this, so we do need to do the percentages to figure out the exact answer. But we do see that there are indeed actually more cases than controls total, but we have more E3, E4 within the case than we do within the control. So, so that could be interesting as well. And so with that, the last part of the notebook here is to just give you some session information down here. But with that, you're able to then export all of these graphs that you've created through running this. Either save it, and you can save it here, or you can copy it and put it into a PowerPoint, or you can actually write some code. You can copy some code over from any of our other notebooks that we've released. There's code in there that will essentially, you can copy it and it'll essentially save this as a PNG to your local or to your VM that's here, and then you can download it that way as well. So with that, are there any questions, I guess, pertaining to the notebook specifically, or the data, or anything like that, or any of the visualizations? Are there, I guess, any ideas for future analyses using this type of data?
Victoria, I just had a question. I think at the beginning of the seminar, you had mentioned that you had like 50 workbooks being produced right now from the AMP-PD community, and I just wanted to know if you could elaborate a little bit on that.
Yes. So it's over 50 notebooks that are being created by the AMP-PD community, and so some of those are grouped into workspaces. Some of those are currently in progress, and others are being worked on by grantees. And so we check in on their progress every once in a while. But it could range from anything like, let's see if we could find the getting started workspace. So something like this, that's a getting started workspace that contains 8 notebooks that are in R or Python. I think this might be an older version of this, but we have, this is an example of a workspace that has 8 of these notebooks. So there are, Start Here notebooks that go into the data. There are some that, take you through merging demographics and doing visualizations. And so these are paired with both Python and R. We also have other ones. So this is from, I guess, previous releases. Let's see if we can find, this is from an older version. So this was from 2020, from a 2020 release. This is looking at RNA-seq, and it's doing RNA-seq QC, and it's also doing a principal component analysis. So what this does is it runs TSNE, and then also runs a PCA, and it has all these different notebooks within it. So this is from an older release, so this isn't counted within that. But we do anticipate people producing, similar things, but also expanding on them within the community workspaces. Barry, Dave, did you guys have anything to add to that?
No, nothing to add. You covered it.
Yeah, so those
I
are,
was on mute again. I was going to say pretty much the same thing. I think you covered it pretty well.
Okay.
Maybe just that the intent here is to have the basic building blocks that any researcher would need, so that you don't have to worry about how do you connect into the data or how do you use the data together. That's already prepared for you, and you can spend your time focusing on your analysis, or even taking somebody else's analysis and building something new from it.
Yeah, so a lot of these are definitely meant as, examples. So like this notebook that we just went through has a lot of code that you could copy into something else. If you're interested in making pie charts, they're somewhere here. You could take this code, right, and copy it into a new notebook, or you can take, your data set of interest, and just instead of, you could leave all the variables the same, but just like replace the BigQuery table here. So this is where you would like change your location, and then you could pull data from a different location. But yeah, these are definitely meant to be examples. You can also take this and expand on it and do more sophisticated analyses. So I did, write this in here. It would be interesting to see if there's any correlations between protein or gene expression, and ApoE genotype and things like that. If there's any differences between cases and control, in terms of, any proteins or genes that are differentially expressed that also correlate with ApoE genotype. That would be a really interesting analysis. That would be something that could be provided by the community if someone within the community wants to expand on this specific case study.
Yeah, thanks for answering the question. That was great. So I guess, yeah, I agree that like the workbooks are super helpful in getting started and kind of expanding on them. I just wasn't sure if when you mentioned like 50 people or 50 groups working on them, I imagine that they'll be published, and I look forward to looking at their analyses and seeing what else can be done. So thank you.
Yeah, no problem. Yeah, and we're so these we're doing a preview release. So like a small data release that'll release some of the targeted proteomics in the new format and the accompanying notebooks. So those will be out very soon. So if you have, I guess for these ones, because we haven't publicly released the data yet, the authorization domain and people who have access to these specific notebooks essentially needed to reach out to me or Willie to be added to the group to access these notebooks. But hopefully within a week or so, you'll be able to just access them if you have tier 2 access and access to AMPD data. So that'll just be a resource that's available. So yeah, so I guess look out for that within the next week or two. But yeah, are you added onto the I guess the beta user group for these kinds of notebooks?
Yeah, I think you actually just added me last webinar. So
Okay.
Thank you for that. Yeah.
Yeah, no problem.
So I was just wondering if there are like protein modifications that are available as part of the proteomics data sets, or is that
Yeah, so
not?
so the data set that we have available right now is a targeted proteomics data set that was provided by Olink. So there is no PTM analysis in that. With the untargeted data set that's going to be provided by Dr. Jennifer Van Eyck's group at Cedars-Sinai. So that's going to be swath untargeted. We will have the raw and the MZML files. I think it's MZML or MZXML. We're going to have those available. So if you want to download them and like search them on your own workflow and look for PTMs, you would be able to do that. However, I don't think the way she's doing the acquisition, it's not specifically targeting PTMs. So she's not doing like a phospho enrichment or anything like that. It is just going to be untargeted data. But if you would want to research it, you would be able to download the data files and do your own searches if you're interested in PTMs.
All right, thank you.
No problem. Are there any other questions related specifically to the notebooks, or I guess just the MPD data in general? Okay. Let me just get back to the slide. Let me find the tab. Okay. So just as a reminder of what we did, we logged into Terra. We found the workspaces section. We found the webinar workspace. We cloned the workspace. In order to share the workspaces, you could also use the same little snowman icon. I went through parts of the workspace and how to set up the cloud environment. I also went through the differences between the edit and the playground mode, right? One saves your changes, one does not. And then actually running the cells and then going through all of the visualizations as well and all of the graphs. Oops. Sorry. And so with that, it would be really helpful, once again, if we get your feedback for these webinars. So there is a survey. It was pasted at the top. So let me just find it and paste it again. If you could fill it out, it'll probably take you about 5 minutes or so. It'll be very helpful in helping us structure any future webinars. If you provide feedback like, maybe you want more technical aspects. Maybe you want something that's more on the fly and like editing code and stuff like that. We can definitely accommodate those kinds of things, but we just won't know, if you want that unless you, put it in the survey. And so, yeah, the survey would be really helpful in helping us kind of maneuver these webinars and make them as useful as possible to everyone. With that, we have 3 more webinars that are scheduled. We have one on May 25th that's going to focus on extracellular RNA expression. We're going to have one on June 8th that's going to look at predictive models of PD risk. And then we're going to have one in August looking at polygenic risk scores. And we're also planning on adding more as more community workspaces become available and come out. And so, yeah, we're excited to have these resources out there. We're excited to showcase them in this format, and we're also really excited to get your feedback just so we can make sure that the webinars are tailored and providing value to you as well. And so with that, it does take a lot of people. This is a very large project, and so thank you to everyone who's contributed. It's definitely it's definitely a team effort to get these large projects going. Were there any questions before we close out? I know that there were some questions in the chat. Does anybody have any questions about if you don't have tier 2 access? Does anybody have any questions about tier 2 access or MPD? data, I guess getting tier 2 access. Are you interested, if anyone's interested in that at all or has any questions about it? Okay. Were there any questions or any suggestions for, I guess, any future webinars? Are there any, I guess, really interesting biological questions that you have of the AMP-PD data?
Yeah, I mean, I guess if you're asking for more general feedback, I'm really interested in sort of genetic analyses and
Okay.
leverage the genomic data.
Awesome. Yeah, we do have quite a bit of genomics data. And our next, so Mary McCarries is going to be doing, I guess, some stuff using polygenic risk scores. I don't know how much of interest that is. I know that there are individuals within the community that are working on using the genomics data. And then, yeah, if you're interested in genomics data, do you have tier 2 access to the AMP-PD data?
Yes.
Okay.
So I've done some analyses, sort of downloading data and then doing it locally, but I haven't worked very much with the AMP-PD framework, the Terra framework yet.
I see. Yeah, the Terra framework is really nice because you don't have to download the data onto your computer or anything like that. And I know some data can be really big. So within using Terra, you can have everything stored in Google Cloud and then just pull tables that way. So that's actually very convenient. So if you have any questions about Terra, feel free to reach out and we can try and help you out as much as we can.
Yeah, I actually have a follow-up question. Oh,
Yes.
go ahead.
Oh, no, I was just gonna change gears. Please go ahead.
Oh, I just had one more sort of question comment, which was that I was wondering if you were planning to introduce any sort of additional resources to help budget and figure out like how much using the resource costs and how much different things cost, because that's one thing I've been struggling a bit to figure out.
That's definitely something we've gotten a few questions about on our admin support email. So yeah, I think that's a great one to add to the list.
Yeah, I know we do have some notebooks here and there, but we don't have anything compiled.
Yeah, I'm not sure. I'm not sure it's just within Terra, but Google Cloud has a number of resources to help you estimate what it'll cost to do certain things, and Terra does have some estimation tools.
Yeah.
So in one of our
yeah,
sessions, we had just real quick. In one of our sessions, we did have a presenter show a cost analysis notebook that's used to evaluate cost of a workflow. I'm not sure that we have anything that can do that same function for notebooks, but we should look into that.
Yeah, I think that's
Also, as
very
a
useful.
follow-up, as a follow-up to that, I think, in terms of billing, it'll be nice to kind of set it up in a way that, like students don't have to really use their credit card, but like invoice, on a monthly or a yearly basis, rather than each user using their own like credit cards for billing purposes. I think that'll make it a lot easier, I think so.
Okay.
So I had a question for the audience. Looking through your webinar series, the way that we've structured these is about members from the community who are working with the data and presenting their work. But would it be helpful to have a basic getting started type of webinar where we're walking through the getting started notebooks? Just how do you access data, more about what generally is in there? How do you query a BigQuery table? Things like that. Would people find that useful? Separate from the these actual analyses.
I think so.
Okay.
Maybe we could have a separate session for that in addition to these.
Yeah, we don't have anything scheduled for June or like end of June or July. So we could potentially schedule something in there. We can discuss scheduling something within that time frame. I think we were trying to keep it open just because of the summer months and people traveling, but if it's a getting started, kind of more informal and not analysis driven webinar, we can definitely schedule something like that during that time.
Also, we have people available to help you with this. If you want to get started on your own and you have questions and need a screen share for a little one-on-one session, we've done that before and are happy to. So you're not alone in this if you want to try doing some of this on your own.
Yeah, so you can feel free to reach out to me or Willie or Barry or Dave, and we can, if we can't help you, we can find someone who can. So yeah, so if you're interested in anything, if you're interested in, we could do a quick call and help you kind of get set up and get started. If you're interested and don't know where to start, we can definitely make time for that.
Wonderful. Thanks so much, Victoria.
No problem. Are there any last minute questions before we close out? Okay. Well, thanks everyone. Yeah, please fill out that survey form. It'll really give us the feedback we need to, make any changes that we might need to make. So those are always really helpful. Thank you so much for attending, and thank you for your questions. Yeah, thanks everyone.
Awesome. Thanks, everybody.
Thanks, everyone.
Thanks.
Thank you.
Thank you.
Bye.